Good morning, students. Welcome to the biology class. So in the last class we learned about the various modifications which are possible in the amino acids. We learned about methylation or otherwise acetylation, phosphorylation, hydroxyl group attachment, disulfide bond formation, and then we learned about some of the properties of proteins. That we understood that proteins are made up of magic 20, and there is a huge variety of proteins around us. Then we learned that some proteins are called first class protein or complete protein, and animal proteins are called as complete protein. Reason, they are able to provide us all the essential and semi-essential amino acids on the daily basis if they are consumed. Plant proteins are incomplete proteins or second class proteins because in the plant proteins generally one or two of any of the essential or semi-essential amino acids would be absent. Soya bean is an exception in which all the essential amino acids are present. Then we learned about that there are huge variety of the molecular weight also. Heaviest protein we learned, lightest protein also we learned. Then we learned about the denaturation property, and then renaturation is also possible if it is a temporary denaturation. So denaturation is of two types, temporary and permanent. When it loses all its bonds and all because of treatment with the strong alkali or acid or temperature or some pressure, then in that case the protein undergoes denaturation. If it is a major breakdown of the bonds, then it could become a permanent denaturation. If it is a minor alteration, then it is possible when the conditions become conducive or correct conditions are there, this temporary denaturation will be overcome by renaturation. That means establishment of the previous bonds again, and the protein becomes a functional one. So denaturation causes non-functioning of the protein. It could be a temporary non-functioning or it could be a permanent one. Then we learned that isoelectric point is also there for every protein, and we learned the example of the curd, that at a pH of 4.7 the protein present, that is the casein protein, the protein present inside the milk is going to coagulate or precipitate, and that's why curd formation takes place. We learned about solubility, that very few of the proteins are soluble, though amino acids are soluble. And then we learned that there are two types of proteins possible on the basis of how many protomeres or how many polypeptide chains are present. If only one chain is present, only one polypeptide chain is present in a protein, it is called as monomeric protein. Example, lysozyme, which breaks down the wall of bacteria, and myoglobin, which is present in the muscles and acts just like hemoglobin. It stores the oxygen. The second category we learned is oligomeric, and we learned that there is no polymeric type of protein. Oligomeric proteins are those which are made up of more than one polypeptide chain, or you can say two chains or more than two chains, and each chain in this case would be called as protomeres. Now we learned the example of insulin and Ig. Ig is immunoglobulin and hemoglobin. Insulin carries two chains, then immunoglobulin carries two chains, and hemoglobin carries four chains. Out of these four chains, two chains are alpha type and two chains are beta type. Then we learned that insulin is made up of insulin in the beginning is made up of an additional chain, that is the C chain, and as long as the C chain remains in insulin, it will be a non-functional insulin, and it is called as proinsulin. So when human body synthesizes insulin, it is proinsulin, and this proinsulin will change afterwards into insulin. So the finally insulin carries only two chains. A chain has 21 amino acids. We learned in the last class that sequence on the arrangement, everything is going to be fixed by the genetic makeup or DNA. So A chain carries 21 amino acids, B chain carries 30 amino acids, and these chains are stabilized by disulfide linkage. So this all we did in the last class. We did a small amount of introduction even to the primary structure, and that's why in the assignment also I gave one question out of this. So the structures of protein. Now the primary structure of protein, we learned that degrees are put for primary, secondary, tertiary and quaternary structure. So the primary structure of protein is made up of nothing but the sequence of amino acids. So amino acids, remember this amino acid sequence, number of amino acid, positions of various amino acid, repetition of amino acids, everything is controlled by the DNA. So DNA will cause the formation of a messenger RNA, and messenger RNA will carry the information like a code, and according to the code particular amino acids will come in a sequence, so there cannot be any mistake. So primary structure is made up of chain of amino acid, like for example a chain of insulin carries 21 amino acids. So this is a fixed thing, this is the primary structure of the chain. Now every chain starts with a left side turn end and the right side end. So there are two ends of this chain. Left side end is also called as N terminal, and the right side end is also called as the C terminal. Now N stands for the, see, because you start with the first amino acid here, so the N group, or you can say the amine group. So amine group is present, or amino group is present towards the left side with the beginning of the chain. That's why this terminal is called as N terminal. And the last amino acid will have its COOH towards the right hand side, and that is called as the C terminal. I hope this much is clear. And then we learned that there is a bond formed between the consecutive, or you can say the adjacent amino acids, and that bond is the peptide bond. And then peptide bond, remember the distance between two peptide bond would be 0.35 nanometer, and remember this is how you represent the peptide bond. Remember in this process one molecule of water is lost. Every time there is a bond formation, which is peptide bond formation, one molecule of water would be lost. And remember this is going to be the first bond, then second bond, it continues like this. When two amino acids join, it is called as dipeptide. When more will join, finally oligopeptide, and this name will continue, polypeptide. Now this much we did in the last class.